Welcome to Excel Class Hub. My name is Dr. Brain, and I'm your biology tutor. We are going to be studying the internal structure of a flowering plant. The internal structure of a flowering plant. All right, so that'll be our topic. First of all, before we go into the details of this topic, all right, it is very, very important that we basically understand some things. Now, first of all, what is a flowering plant? What is a flowering plant? Now, recall that the plant kingdom, the plant kingdom is basically divided into three. The plant kingdom, usually called the kingdom plantae, is basically divided into three. All right. We have the Talophyta, we have Bryophyta, and the Tracheophyta. So these are the three major divisions of the plant kingdom. All right. So under your Talophyta is where you have an organism such as your Spirogyra. Your Spirogyra belongs to the Talophyta, your Spirogyra. Then under your Bryophyta is where you have your organisms such as mosses and liverworts. Mosses and liverworts. Or as a means of actually expanding these things, okay, let's do a detailed classification of all these things so that it's going to be very, very clear. So, but now we said the plant kingdom is divided into Talophyta, Bryophyta, into Bryophyta, and into Tracheophyta. Yes. So those are the three major divisions of the plant kingdom. So talking about the Talophyta. The Talophyta are also called algae. They are also called algae. And they are further divided into three classes. We have chlorophyta. We have rhodophyta, and we have phycophyta. So these are the three major classes under the division Talophyta. Now recall again that I said the Talophyta are also referred to as algae. Now, of course, from your knowledge of biology so far, you should know that anything that is called chloro means green. It means green. So chlorophyta means green algae. Rhodophyta, rhodo from the word red. So rhodophyta means red algae. Then your phycophyta means brown algae. So that is the Talophyta. Now talking about the Bryophyta. Bryophyta is further divided into three classes. Bryophyta is further divided into three classes. We have class Musi. That's the first one. E.g., this is where you find your mosses. Class Musi. E.g., Moses. Then the second one is class Hepatica. So an example of an organism here is your liverworts. Class Hepatica, e.g., liverworts. Then the third one is class Anthoceratae. class Anthocerote. An example of an organism here is your onwards. Onwards. So the bryophyta, the division of the plant kingdom called bryophyta, is further divided into three classes, which includes class Muci, e.g. Moses, class Hepatica, e.g. liverwort, class Anthocerote, e.g. onwards. So having said that, now, so we move to the third one. The third one is the tracheophyta. The tracheophyta. Now, these tracheophyta, they are commonly called the vascular plants. They are commonly called the vascular plants. And why are they called vascular plants? They are called vascular plants because they have conducting vessels for the conduction of food and water. So the tracheophyta is now further divided into two. We have pteridophyta. Pteridophyta. This is where you have your organisms such as ferns. Ferns belong to this category, pteridophyta. Then the second one is your spermatophyta. Spermatophyta. Now, spermatophyta are also called seed-producing plants. These are plants that produce seeds. It is only when we get to spermatophyta under the plant kingdom that we start seeing production of our seeds. Plants existing before spermatophyta are, for instance, in bryophyte and pteridophytes, that is the plants before the spermatophyta, what they basically produce is spores. So those categories of plants basically produce these spores. But the production of seeds begins from the spermatophyta. And that is why spermatophyta are called the seed-producing plants. And this spermatophyta is now further divided into two. All right? The seed-producing plants are further divided into two. We have the gymnosperms. The gymnosperms and the angiosperms. The gymnosperms and the angiosperms. Gymnosperms are plants that have naked seeds. They are plants that have naked seeds. And these naked seeds, these naked seeds are usually seated or borne on special structures called cones. But however, these angiosperms are otherwise known as flowering plants. And that is exactly where we are going. Angiosperms are otherwise known as flowering plants. Flowering plants. And that is our major concern. And of course, they are the most advanced categories of plants. They are the most advanced categories of plants. So, when we talk about flowering plants, we are talking about the angiosperms. The angiosperms. Angiosperms, also called flowering plants. Flowering plants. So another name for flowering plants is angiosperms, the most advanced categories of plants. And these angiosperms, they are now further divided into two. We have the dicotyledons, which is the most primitive of both. Then we have the monocotyledons, dicotyledons and monocotyledons. Dicotyledons are more primitive than monocotyledons, while monocotyledons are more advanced than dicotyledons. Monocotyledons are more advanced than dicotyledons. So what are we saying in essence? Flowering plants are also called angiosperms. Why are they called flowering plants? Apart from the fact that they are seed producing, they also bear flowers. They bear flowers. That's why they are called flowering plants. All right. So apart from the fact that they are actually seed producing plants, they are also characterized by the presence of flowers in them, right? By the presence of flowers. And of course, it is in flowering plants that you begin to see structures such as floral parts. That is the part that makes up the flowers, the floral part, such as your petals, your sepals, uh, your stamen, your pistil, and so on and so forth. So now, having established what a flowering plant is, so now we can now begin to examine the internal structures of flowering plants. Can now begin to examine the internal structures of flowering plants. So first of all, like we said, that flowering plants are divided into monocotyledons and dicotyledons. So that means that if we are going to be studying the internal structures of a flowering plant, we then have to study the internal structures of both dicotyledons and monocotyledons. Now, and when you look at flowering plants, you notice that the bodies or the body of a flowering plant is basically divided into three parts. The first one is the roots. The second one is the stem. And the third one is the leaf. So when you look at a flowering plant, you are going to see these three distinct regions. All right. We are going to be seeing the roots, the stem, and the leaf. So when we want to study their internal structures, that means we definitely have to study the internal structure of the root, the internal structure of the stem, and the internal structure of the leaf, of the leaf, of the leaf. This is very, very important. This is very, 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 very important. So now, having established that, and again, like I mentioned earlier, that flowering plants are divided into two, dicotyledons and monocotyledons. So as we are studying the internal structures of a dicotyledon, we also study the internal structure of a monocotyledon under these three headings: the roots, the stem, and the leaves. All right. Now, how do we study the internal structure of a flowering plant? We study the internal structure of a flowering plant by taking a transverse section of that particular part that we want to study. For instance, if we want to study the internal structure of the roots of a flowering plant, the first thing we do is that we take a transverse section of that particular part, e.g., the root, for instance. Then after taking a transverse section of it, we stain with a dye. All right? So, and why do we have to stain with a dye? We normally stain with a dye so that structures that ordinarily are not supposed to be visible will become visible. In other words, there are some certain structures internally in a flowering plant that ordinarily shouldn't have been visible, but only becomes visible after staining with a dye. So after taking a transverse section of that particular part of the plant, we stain it with a dye, then observe it with either a hand lens or a microscope. So after taking a transverse section and staining with a dye, we now use either a hand lens or a compound microscope to view those particular parts. All right. So having said that, so first of all, like I told you the other time, flowering plants are divided into two, dicotyledons and monocotyledons. The dicotyledons, like I said, are the most primitive. They are the most primitive, while monocotyledons are the most advanced. So that means that we are going to be beginning our study from that of the dicotyledons. And remember, I also told you that there are three parts we are going to be studying. We are going to be studying the internal structure of root, of stem, and of leaf. So let's begin with the internal structures of a dicot leaf. Of a dicot root, I mean. The internal structures of a dicot root. Internal structures of a dicot root. By dicot, I mean dicotyledons. Yes. So what I mean by dicot here is dicotyledons. So we want to look at the internal structures of a dicotyledonous root. Of a dicotyledonous root. Now, take note of this. In the internal structure of a dicot root, from the outside to the inside, these are the structures we are going to see. The first one is what we call the piliferous layer. The piliferous layer. The second one is the cortex. The second one is the cortex. After the piliferous layer, we have the cortex. Then after that, we have what we call the endodermis. Endodermis. The fourth one is the pericycle. Pericycle. Then the fifth one is the xylem and phloem. The xylem and phloem. So these are the parts of the internal structures of a dicot root. So from the outside to the inside, these are the structures we are going to see, beginning from the piliferous layer to the cortex, from the cortex to the endodermis, from the endodermis to the pericycle, from the pericycle to the xylem and phloem. Also, now take for instance, how do we differentiate, first of all, the internal structure of a root from that of a stem? How do we differentiate the internal structure of a root from that of a stem? Now, you notice that under exam conditions, the internal structures of both roots and stems are usually represented in the form of a circular structure like this. In the form of a circular structure. But however, how do you differentiate the internal structure such that when you see the picture, how will you know immediately that this is a root and not a stem? Now, we normally differentiate the internal structure of a root from that of a stem by the presence of what we call the root hairs. The root hairs. So immediately you see the circular structure, and you want to be really sure if that structure is the internal structure of a root or the internal structure of a stem. The first thing you look out for is the appearance of root hairs. The appearance of root hairs. So if you can see the appearance of root hairs in that particular structure, then that structure is definitely a root. Definitely a root. But if root hairs are lacking in the structure, then that particular structure is a stem. Because every root, whatsoever we call the internal structure of a root, must possess root hairs. Must possess root hairs. That's settled, that aside. So like I was saying, if we examine the internal structures of a dicot root, these are the structures we are going to see. The first one is the piliferous layer, followed by the cortex, followed by the endodermis, followed by the pericycle, and followed by the xylem and the phloem. Xylem and the phloem. So now, the piliferous layer, take note of this. The piliferous layer is also called epiblema. The piliferous layer is also called epiblema, or you call it the rhizodermis. So another name for the piliferous layer is the epiblema or the rhizodermis. Epiblema or rhizodermis. So after the piliferous layer, we now see the cortex. After the cortex, we see the endodermis. After the endodermis, we see the pericycle. Then after the pericycle, we see the xylem and the phloem. So let's begin by talking about the piliferous layer. Let's begin by talking about the piliferous layer. So now, let's represent the internal structure of a dicot root by this circular diagram. So like I told you earlier, provided it is a root, you are going to see the appearance of root hairs. That's certain. So you must see root hairs. So I'm trying to sketch my root hairs like this. For instance, so let's assume that those are root hairs. Then you see some structures inwardly. Now, we are still going to draw the diagram in a more expanded way so that we can see the structures that follows the piliferous layer. But this particular layer that you are seeing, from which the root hairs arises, is what we call the piliferous layer. This particular layer where the root hairs arises is the piliferous layer. Yes, like you can see, these are the root hairs. So, what are we saying in essence? The piliferous layer is a single layer of rectangular cells from which the root hairs arises. Just like you can see from the diagram, the root hairs actually arise from this outside external layer called the piliferous layer. Immediately after the piliferous layer, now this empty space that you are seeing before the other remaining structures is the cortex. Yes, the cortex. So now, like I said, the piliferous layer is a single layer of rectangular cells from which the root hairs arises And again, it is also pertinent to understand that the piliferous layer lacks what we call cortical. The piliferous layer does not have cortical. Now, cortical is found in stems and leaves. And that's why you notice that the external layer of the internal structure of a stem and a leaf is mostly called epidermis. But epidermis is not usually used for roots. Why? Because the external layer of the internal structure of a root is called piliferous layer and not epidermis, because the external layer of the internal structure of a root does not have cortical. What does cortical do? Cortical basically does protective functions. Now, cortical is just like a waxy layer that prevents water loss in stems and in leaves. So the function of cortical is that it actually prevents water loss. But a root cannot have cortical. Why? Because the root hairs that are associated to the piliferous layer of a root, the functions, in fact, the major function of the root hairs is absorption of water and mineral salts. So if a root should have cortical, then the function of absorbing water and mineral salts then practically becomes impossible. Because if there is a layer of wax that is blocking the absorption of water by the root hairs, then it creates a problem for the plant. So that is why the reason why the piliferous layer does not have cortical, unlike the external layer in leaves and in stems, is because the root hairs are associated to the piliferous layer. They arise from the piliferous layer. And one of the functions of the root hairs is the absorption of water and mineral salts. So if cortical is located there, then the function of absorbing water and mineral salts then will practically be impossible. So that is the reason why cortical is not present in the piliferous layer of a root. Also, it is important that I also state that this piliferous layer is not a permanent structure in the roots, in the internal structure of the roots. Now, as the plant begins to grow, this piliferous layer will begin to break away from the plant. The piliferous layer will begin to break away and will be replaced by a layer called the cork layer. Cork will be replaced by the cork layer. It's replaced by what we call the cork layer. So the piliferous layer is not a permanent structure in the internal structure of the root, because as the plant ages, the piliferous layer will now be replaced after breaking away by what we call the cork layer. So now, having said all that, what then is the function of the piliferous layer? What then is the function? What then is the function of the piliferous layer? What then is the functions of the piliferous layer? What then is the function of the piliferous layer? The first one, now recall that we said that the root hairs are associated with the piliferous layer. That is, the root hairs arises from the piliferous layer. And what is the function of the root hairs? Absorption of water and mineral salts. So, meaning that the first function of the piliferous layer is the absorption of water and mineral salts. Absorption of water and mineral salts. That is the first function of the piliferous layer. And how does it absorb water and mineral salts? With the aid of the root hairs that arises from it. So, in other words, the piliferous layer helps in the absorption of water and mineral salts with the aid of the root hairs that arises from it. The second function of the piliferous layer is that the piliferous layer protects the underlying cells. Now recall that I listed about 5 structures that can be found in the internal structures of the dicot roots, of which the piliferous layer is the first layer, the external layer. So apart from the fact that the piliferous layer helps to absorb water and mineral salts, one other functions that it performs is that it serves protective functions for every other cells that are inside or that it surrounds. For instance, you notice that after the piliferous layer is where we have the cortex. After the cortex is where we have the endodermis. After the endodermis is where we have the pericycle. Before we now have the xylem and the phloem. So all these structures that are inside the piliferous layer helps to protect them. So that's what we mean by the fact that it protects the underlying cells. It protects the underlying cells that are inside the piliferous layer.